actually what I'm going to do, mount it in here first, and then swap out with you.
Now you're adding media.
So now I'm going to aspirate. I'm going to try and be as gentle as possible.
Okay, a few seconds, just hold it.
Okay. So the important features of our live cell imaging grid are that, so it's an inverted scope, which means the objective, the sample is here and the objective is underneath. So the objective is pointing up. And the whole scope, or practically the whole scope, including the objectives and the sample, are at 37 degrees. So everything in this plastic box is at 37 degrees. And right on top of the stage